a good crowd already. Okay, so let me just introduce Kumar Narayanan from the University of Iowa. Kumar is a physician scientist in Iowa City. He is the Bartlett Professor of Neurology. He's also the director of the University of Iowa P30, which is kind of a Udall Center precursor. I had to say I had the considerable pleasure of working with Kumar on a project over the last year and a half, which is that he and I co-edited a book, which I think is a pretty good book, but most important, I really enjoyed working with him on this project. So he's going to talk today about the very interesting work that his group has done on Parkinson's disease and cognition.
Thank you, Roger. And I have to say this talk was a little bit of a challenge because Roger invited me to talk in 2020, just before the pandemic. And so I tried to replace most of the slides with data that Roger hasn't seen. So just to keep it interesting. The title of my talk is Prefrontal 4 Hz Rhythms and Cognitive Control in Parkinson's Disease. And if you don't recognize the image in the back, you're not a fan of 90s rap. I think only Dan Leventhal will recognize this image, which we'll talk about in a bit. In the 90s, there's a rap group called Tribe Called Quest, and this is one of their album covers. The album was called People's Instinctive Travels on the Paths of Rhythm, one of my favorites in 8th grade. Briefly, my disclosures is funded by NIH, the University of Iowa, the Defense Department, and private philanthropy. Most importantly, oops, I went too many slides here. I want to start by talking about one of my patients. This patient is a patient at the VA that I treated as a resident. And here I am going through the UPDRS. And you notice he's in a seatbelt. We're about to put him in a nursing home. He had profound cognitive symptoms of Parkinson's disease. And in some sense, my entire career and the things we do at the University of Iowa is trying to come up with better treatments for patients like this. And we'll return to his movements in a second. But in going through the Unified Parkinson's Disease Rating Scale, we noticed that he was incredibly variable in time. And so I'll come back to that in a bit. As we all know, I don't have to tell this group, Parkinson's disease, the cardinal symptoms are tremor, bradykinesia, rigidity, and postural instability added by Charcot. But I don't also have to tell this group that there are all kinds of non-motor symptoms of Parkinson's disease that we all spend a lot of time in clinic wrestling with, including cognitive dysfunction, dementia, psychosis, impulse control, and mood disorders. And while we know some things about the motor symptoms of Parkinson's disease, I would argue that these symptoms of Parkinson's disease, which are defining for a patient's trajectory, we don't know much about the circuitry and we don't have very good treatments. There are 3 broad classes of mechanisms that we could debate. One is that cortical Lewy bodies contribute to cognitive dysfunction in Parkinson's disease. As everyone here knows, Parkinson's disease involves toxic proteins such as alpha-synuclein, maybe amyloid. Alpha-synuclein can end up in the midbrain dopaminergic nuclei. It can end up in the basal forebrain. It can also end up in the cortex. Another possible pathophysiological theme is that there are these massive ascending projection systems, ventral tegmental area, substantia nigra, basal forebrain, locus coeruleus, serotonin neurons that project all over the brain, and it's dysfunction in these projection systems that lead to or contribute to cognitive symptoms of Parkinson's disease. And a final mechanistic theme is that there are genetic mutations such as ApoE4, Gaucher syndrome. We could pick a number of these mutations that contribute to cognitive symptoms of Parkinson's disease. I would, I'm not going to speak for you, but I'd say that both of us, both groups, Iowa and Michigan, we have placed these neurotransmitter systems at the forefront of the way we study the disease. Your group has been sort of absolutely pathfinding and pioneering in acetylcholine. And the advantage of these is that they have circuits, they have themes, and they have interventions. So I will talk about one of these projections, the dopaminergic, the VTA dopamine projection. My goal today, and actually it's the same goal, Roger, that I had 2 years ago, and potentially the same goal I'm going to have for the next 20 years, is to convince you of this single statement. Dopamine is required for 4 Hertz cortical rhythms controlling cognition. And I'm going to do this by talking about patients with Parkinson's disease and how they have attenuated 4 Hertz rhythms. We're going to try to take a deep dive into the rodent mechanisms of cognitive control. And then we'll talk briefly about some stimulation data at 4 Hertz and our efforts to improve cognitive control in Parkinson's disease. Now, I can talk a lot more about each of these topics, and I encourage you to interrupt me. I know, I would hypothesize this is a spirited group. I've had conversations with many people over the years, and so please interrupt me, quibble, ask questions, challenge me. We can make this Zoom call fun. All right, we'll take the first theme here. Patients with Parkinson's disease have attenuated 4 Hertz rhythms. So in order to do this, I'm going to turn to a very old technique in neurology, electroencephalography. This technique, as everyone knows, about 100 years old, used heavily in neurology for seizures, but I think has a lot of diagnostic potential. And it has that potential because, as opposed, but, PET, MRI, those things require a significant amount of resources and are expensive and cost a lot of money and often cannot be done continuously. Whereas EEG is ubiquitous, cheap, and can be done continuously and can be used as a closed loop control signal. So I think EEG has a lot of potential. It has very poor, horrible spatial resolution, centimeters, if that, but it has excellent time resolution. And so we're going to take advantage of that. If I was to play a loud sound, and we can't do this over Zoom, but this is a 110 decibel sound equivalent to a train passing by, and we were to place a recording over scalp electrode CZ, lowering my head so you can see where it is at the vertex, you would see reliably a 4 Hertz oscillation that was triggered by that sound. And, neurologists have been looking at this signal in the voltage domain, meaning the y-axis is time and the x-axis is voltage, but we can actually look at this signal in the frequency domain. Here, the y-axis is frequency and the x-axis is time. And when you look at it in the frequency domain, it potentially becomes much clearer. There is a 4 Hertz burst that's triggered by this loud sound. This 4 Hertz burst, and I told you EEG has horrible spatial resolution, but it seems to be centered over the mid-frontal or medial frontal cortex. And, we did this study in here at Iowa, but the person who's really advanced this idea is a guy named James Kavanaugh from the University of New Mexico. And he's sort of written many reviews on the meaning of the signal, as well as kind of 90 original research articles. So he has a lot to say about what this signal means, and I'll try to give you some of that insight. But sort of the departure point for this talk is if we look at Parkinson's patients, demographically matched Parkinson's patients, these controls play the same sound, we notice that this rhythm is markedly attenuated. We can see it in the voltage domain, but it's much more apparent if we look at it in the time frequency domain, in the frequency domain. So, these Parkinson's patients seem to be missing this rhythm. And, one thing about my lab, we've done this every which way to Tuesday, but, I worry that something about our analytic technique or so on, but this technique, this finding has been replicated kind of independently. And I believe in it strongly enough that if you do this, I think you'll see this as well. So we think that this rhythm is really, consistent in patients with Parkinson's disease. You'll see an attenuation in this 4 Hertz rhythm in response to loud sounds. So as I told you, Jim has really spent the last 10 years since he started this lab. He came from Michael Frank's lab at Brown and went to New Mexico. He's really characterized this 4 Hertz burst. So every time you see things that require cognitive control, novelty, conflict, punishment, errors, you will see a 4 Hertz burst of activity from the midfrontal cortex. This is kind of a meta-analysis over several different tasks. And yeah, there's slightly different topography, slightly different frequency bands, but it's remarkably consistent. And Jim has suggested, and this is really his theory, that this 4 Hertz rhythm, and by this I mean theta. Now, the reason I didn't say theta is that 4 Hertz is right on the artificial band between delta, which is 1 to 4, and theta, 4 to 8, 5 to 8, depending on the definition. And that kind of reflects the problem that we have with our spectral approaches. These bands don't really describe. This is right on the cusp of these things. And you can see that this frequency burst really spans those two bands. So he has argued pretty convincingly that this is a cognitive control signal. This signifies the need for cognitive control. And then work in our lab is really kind of trying to illustrate how deep the rabbit hole goes. So I'm going to give you, this is a complicated figure, it just came out in cerebral cortex, but I wanted to walk you through it because it kind of gives me a sense, even at a low end, of what this signal can mean. So here we are recording in the operating room human single units. Every time the human subthalamic nucleus neuron spikes, you see a tick. And this is the center channel EEG. Now it's not quite CZ. We're in the operating room. There's a stereo field. For some reason, the neurosurgeon won't let you let it out with alligator clips and try to place things with. So Jeremy Greenlee's our neurosurgeon, enormously collaborative, pleasure to work with, and has really entertained our hypotheses. So he put this channel pretty close to the vertex, played an auditory stimulus, and here's the response. This is a response modulated neuron. This neuron is firing right up to the response. But what we notice is this 4 Hertz rhythm at the time of the auditory stimulus, the STN neurons tended to fire in phase with this 4 Hertz rhythm just for a few spikes. If we look across our ensemble, I think we have 54 neurons. This is of the 17 neurons that were response modulated, we saw marked sort of what we call, this is called spike field coherence. So this suggests that this 4 Hertz rhythm could entrain and engage neurons deep in the basal ganglia. And so it provides me with this kind of insight into kind of how detailed the system could go. And this is, again, not just shown by us. Kareem Zaghlul's group has shown this a couple of different times, and we've shown it a couple of different times. So I think it's kind of a pretty consistent finding. Again, these 4 Hertz rhythms recruit single neurons, even deep in the basal ganglia. Sorry, I put a red box on it to make it clear. And so we're kind of studying this in both our operating system, and we're also, this is Brooke Yeager, a new grad student in the lab, who is now looking at the PPMI data set, and her goal is to link this 4 Hertz rhythm with kind of resting state connectivity and functional connectivity, both in PPMI data that we collect, and see if this 4 Hertz rhythm projects on the patients with mild cognitive impairments. So I showed you data with Parkinson's disease, and all of you notice that Parkinson's patients have cognitive deficits. And so for the last 5 years, we've had an R01 to study this 4 Hertz rhythm in Parkinson's patients with impaired cognitive control. And we did this using a suite of tasks that we knew involved cognitive control processes. These are all classic tasks. There's a Simon task in which there's cues and you respond. Sometimes the cues are congruent, which is like respond left on the left side, or sometimes they're incongruent, respond left presented on the right side. There's an oddball task in which there's normally a standard cue and then, respond left or right, but about 20% of trials, there's an oddball, which is a novel stimuli you've never heard before, like a bird call. And then there's my favorite, the interval timing task, and I'll tell you why it's my favorite later on. Again, a very simple task. These are like, estimated interval of 7 seconds, estimated interval of 3 seconds. Now, I could get very fancy and very complicated with these tasks, but these tasks are all designed such that they could be studied potentially in mice, and they are compatible with intraoperative physiology. And so the data I showed you just now is from the oddball task. It's a challenge to do physiology in the operating room. I can't get patients to do tasks that are very complicated, so I try to keep it simple, and that's why we pick these tasks. So, and this finding is just amazing to me, so I'll walk you through it. So what we did is about 45 controls and about 92 Parkinson's patients. They're patients with dementia, patients with mild cognitive impairment, and here we're using Littman type 1 screening criteria, just the MoCA, to separate these patient groups. And you can see here that I told you that Parkinson's patients have less 4 Hertz rhythms, but it's really the patients with dementia in all tasks that have less 4 Hertz rhythms. And the patients with cognitive impairment are kind of in the middle. And then the Parkinson's patients who are cognitively normal are in the range of controls. So this is, to me, a breathtaking finding, because if I told you, there was an EEG signal that we could do from a simple task to predict who has cognitive impairment, maybe it wouldn't help people at the University of Michigan, one of the best places in the world, I'd argue, for Parkinson's research, but it might help people in, I could just pick on any Midwestern town, right, where there are neurologists, or in some case, primary care doctors, who likely aren't spending a lot of time thinking about who has cognitive impairment in Parkinson's disease. And the nice thing is that we had all of our patients, these are the same patients, do the Simon, oddball, and interval timing tasks, and we can compare them head to head. And so in order to do that, we did, so this is a straight correlation matrix. This is a little bit of a joke because the color scheme of JET is exactly the color scheme that A Tribe Called Quest used in 1991. Only probably Dan Leventhal gets this joke. Dan, are you a 90s rap fan?
Not especially, but I still get it.
Okay, that's important. Anyway, so we can do a correlation matrix where we can correlate every single one of these values with the congruent theta versus the oddball theta versus the interval timing theta. And we can use techniques like principal component analysis to try to reduce the dimensions of this, so we can reduce this very complex data set to one number, which is beautiful because PC1, explaining upwards of 35% of the variance in our data set, seems to load very highly on the delta and theta values across tasks. I'll point out, this is, again, because it's data I didn't want Roger to have seen before. This is preliminary data, so it's basically straight from MATLAB. Early days, I'm still error checking it and going over it, but really exciting for us. But what was even more exciting for me is that if we looked at principal component 1, and this is basically, you can say the average theta loading, it is highly predictive of dementia and Parkinson's disease, and it can identify the patients with dementia or patients without dementia, and it seems to really correlate with their cognitive status. So, and to convey my excitement, I did not pre-register this idea because in 2017, when I submitted the grant, I had no idea how to do that. Now I do, but this is what I put in my grant. This is figure 1C from page 76 of my R01 that we set out to do. And the reason I put this up, this is remarkable. Whenever I have hypotheses, normally the answer ends up being way more complicated than I thought. But here on the y-axis, I put delta theta power and controls PD, MCI, and I wondered about dementia patients, and we ended up with that relationship, which is remarkable. So for the first time in my life, something ended up the way I thought it would, which is cool. And then not surprising that because they're defined based on the Montreal cognitive impairment, there's a strong prediction with MoCA. But if we take performance from the NIH toolbox of another task, the Erickson flanker task, which is a measure of cognitive control, we see a very strong relationship, about 0.47, between the Erickson flanker performance and PC1 based on delta theta activity. Finally, we can gussy up machine learning algorithms to try to predict based on EEG who has Parkinson's dementia and who doesn't. And our algorithms actually, tend to, first of all, consistently outperform chance, but they tend to get upwards of 70 to 60% right in identifying these dementia patients and identify patients who don't have dementia. Of course, our algorithm falls apart when we look at mild cognitive impairment because, as you would imagine, the mild cognitive impairment group is intermediate between the dementia and the non-dementia patient population, as you'd expect. So anyway, I thought that was an exciting finding and demonstrates that 4 Hertz rhythms are impaired in Parkinson's disease. But I wasn't quite satisfied. So, like Iowa, Michigan like Michigan, Iowa's a Big Ten program. We have, really good engineers. So I walked across the river. I asked our engineers to ask them how they would approach the problem. And they came up with a kind of very creative, way to go about this. They tried to use an autoregressive Ewell-Walker estimation of the power spectra. And I'll just give you, this is the example of Ewell-Walker from MATLAB. That's actually from the MATLAB help, so you can make fun of me about that later on. And they just basically fit the power spectra. And you notice that this is a very holistic way of looking at power spectrum. The way neurologists look at things, if you do QEEG, on the y-axis, you get frequency bands, on the x-axis, you get time. So they're not going to make any assumptions about bands, times. They're just going to try to fit the power spectrum and try to come up with, parameters to explain the fit of the power spectrum. This is actually an extraordinarily fast technology. It's called linear predictive coding. It's based on speech compression. So basically, the goal is to compress power spectra to a few numbers that you can expand with an autoregressive series. It's exciting because it's compatible with real-time analysis. So, if you wanted to do the MoCA 3 times in a patient, you couldn't do it, right? Because there would be practice effects. Patients would have fatigue. But with EEG, you kind of get this dynamic, continuous measurement that you could imagine to get over the course of a day, several times a day in a patient. And what these guys, we basically term this algorithm the LEAP-D, which is the linear predictive algorithm for EEG analysis of Parkinson's disease. The LEAP-D score, again, a single parameter expressing an autoregressive series, has this almost spooky correlation with the Montreal Cognitive Assessment. So, we're kind of working. So Fahim Anjum and Saurabh Dasgupta, who did this analysis, we basically showed that we can identify control versus Parkinson's in previous papers, and now we're kind of applying this to cognitive impairment in Parkinson's disease. And this is based on 8 electrodes and 5 minutes of EEG data. So it's a very highly accurate, Spearman's row, very highly accurate technique, and we're kind of excited to develop this going forward. This has no mechanistic insight because this is just machine learning. You're basically trying to get the best correlation you can. And whereas if we went through a strictly mechanistically derived approach, we end up with these 4 Hertz rhythms. We're able to raise these 4 Hertz rhythms to cognitive control. Here, I don't know what's going on other than LPC fitting of the algorithm. If we look back at the algorithm, you can see that the theta band tends to be the biggest contributor to the LPC algorithm, but the machine learning algorithm will be different every time depending on how you optimize it. Okay, so hopefully I've convinced you or provided some evidence that patients with Parkinson's have attenuated 4 Hertz rhythms. Maybe I convinced you last time of this, but this is way better data. I didn't have, this project was ongoing. I didn't have so many patients and I didn't have so many conditions last time. So, as a basic scientist, and primarily that's kind of my training, what I've shown you at best is correlation. And really what I want to know is mechanism. And so this is sort of the question that animates our lab. What are the mechanisms of these prefrontal 4 Hertz rhythms? So in order to get at we're going to go at one aspect of this task, these tasks. We're going to go at the interval timing task. So to review, this is a task where patients have to estimate an interval of several seconds. And if you think back to that video of Milton, actually, because this is Zoom, I can go back there and we can watch Milton's movements again through this lens. Okay, so here Milton is going through the UPDRS and I'm giving him my collaborator is giving her a rhythmic stimulus, but he has difficulty following the rhythms, particularly on his left hand. And if you're a motor aficionado, you could say, well, yeah, the tremor's kind of convolved with his sort of rhythmic movements, and that's what's going on. But I would argue that he has a greater variability in his statistics of movement. And so that was the finding, that variability in the statistics of movement, I really tried to put into sort of a quantitative framework. And that's why I turned to the interval timing task. Sorry, I don't want to give you Zoom vertigo here. So, it's been long known before I started this work that Parkinson's patients have impaired timing. But what we noticed is this is not what you might predict in medical school. In medical school, or if you read Evarts's papers from the 80s, you might predict that Parkinson's patients are slow. But what happens is Parkinson's patients actually have something more interesting is going on. They have greater variability in their estimates of this 7-second interval. Whereas control patients have a sharp estimator. I asked them to press the button when they start estimating the 7-second interval would have elapsed, so which is why they start just before 7 seconds. Parkinson's patients are more variable. And we can quantify that actually. We can look at the coefficient of variation. And again, not a small amount of patients. If we look at controls versus patients with Parkinson's disease, their coefficients of variations are consistently higher. And I'm not doing a good job calling out the people who did the work. This is work by Arun Singh. He actually led the big study of cognitive control. He's now an assistant professor at the University of South Dakota. Arturo Espinosa, one of our star technicians who did all this work, and Rachel Cole, who is on the job market in 2022. So, we have shown previously that Parkinson's patients had increased variability in interval timing. Here we showed it with a much larger sample. But amazingly, if we put the Montreal Cognitive Assessment on the x-axis and we put the CV on the y-axis, and again, I apologize, this being straight from MATLAB, but I got to keep Roger guessing with some new data. We see this, again, very strong correlation. And I'm going to go as far as making the statement, and I only have the courage to make the statement because it's on Zoom. If this is in person, I'm sure someone would throw something at me. I think that this is the best correlation of any behavioral paradigm with cognitive function that I can think of. Now, you may know something, and I would be willing to consider, but maybe the Wisconsin card sorting task. But this is pretty high for an orthogonal. There is no timing aspect of the Montreal Cognitive Assessment. There's nothing that involves timing on that test. But here we have this sort of orthogonal measure. And I'd be willing, if you have other opinions, or I'm willing to entertain and shoot me down. But it's pretty high. Now, humans are good at timing, but rodents are the best. This is a desert kangaroo rat evading a snake. Here, this is a classic reaction time task, but you can see that, as if you've ever tried to catch a squirrel in your backyard, rodents are, pretty good at timing. A mouse in your kitchen will out-time you. This is, I would argue, mammalian killer app. This is how we beat the dinosaurs. We flexibly guided our movements in time. So, obviously we can't record on these animals, but we could record in a kind of more structured setting. This is in the operant arena. We can design an interval timing test that's really compatible with mice. So I'm going to walk you through this task because a lot of data will come from this task. So the mice make a nose poke on the back wall, and then they go over to the short hopper and they make nose pokes. On half the trials, they get rewards for the first nose poke after 6 seconds. We call those short trials. We throw those trials out. We don't analyze those trials. On half the trials, everything is identical. The animals start nose poking, but they don't get rewards for nose pokes after 6 seconds. They have to switch to the other nose poke, and they get rewards for the first nose poke after 18 seconds. So what tends to happen is the animals nose poke here for 6 seconds, and they realize they're not getting a reward. They have no cue. They just have their internal estimate of time, and they realize that they got to switch to the other hopper, or the other nose poke to get a reward. And they typically start making nose pokes well before 18 seconds. We call the time the last nose poke at the 6 seconds the switch trial. It is a measure of animals' internal estimates of time. So in order to make this much clearer to you, I thought I would illustrate. This is Alex Bova. Now, no deep lab cut here, just straight up video. So here, animal initiated trials of last back nose poke. It's futzing around. There's some cues. Realize that 6 seconds is up. Went to the other nose poke. Checks for food, and then we'll get a reward here in a second. And so you can tell that this animal clearly understands the temporal statistics of the task. Knows when food is supposed to be available, when like a nose poke at the short nose poke doesn't lead to anywhere, and when a nose poke at the long nose poke is more likely to get rewards. And there are all kinds of interesting things out here. This task comes from the foraging literature. It was developed by a guy named Fuad Balchi and Randy Gallistel, and can be used in all kinds of interesting ways for foraging. You can change the probabilities of reward and so on and so on. We like this task because we can go to town on the models of, we can use our models to dissect these circuits in great detail. So the first one we went after are the mesocortical circuits containing the neurotransmitter dopamine from the ventral tegmental area going to the frontal cortex. This is from the Allen Brain Atlas. I just picked it up as a kind of image. And the technique we love, because it's a sledgehammer, is dopamine depletion. Here are the humans, data I already showed you. Parkinson's patients have increased variability. So we depleted dopamine from the, from the NIGRA and the VTA. We have more data on this now. It's still evolving. And what we found is if we deplete dopamine from the Niagara, we don't change the variability. We do change the switch time, meaning the animals take longer to switch, but we don't change the variability. But if we deplete dopamine from the ventral tegmental area, we see increased variability. And this finding, to me, it's a little bit amazing, even though, there's a lot of caveats you might throw at this, because here we have what I think of one of the first models of what's going on in a patient like Milton. We take away dopamine from a single brain area and we increase the variability of their movements. So we've shown this several times in several different ways. This is the clearest expression of that. You might ask, well, is it the dopamine depletion? So I, in 2012, did RNA interference against tyrosine hydroxylase. We've done some optogenetics. We've done, we blocked the postsynaptic receptors in the frontal cortex. So it's really been the focus of my lab, this single finding. But, I hadn't really linked it to the human literature until we, did the variability of the interval timing task. So, and I throw the slide up there because I've given enough talks where someone will raise their hand and say the VTA is not involved in Parkinson's disease.
No, it's not true.
Right. Well, we looked in 2016 because I got enough grant reviews, and Stephanie pulled every case in literature to add 3 of her own. VTA is involved. It's not as involved as the Niagara, 67% cell loss, but it is involved in Parkinson's disease. And if you look back, Kurt Jellinger and Javoy, people have shown this time and time again. So bringing back to 4-Hertz rhythms, here is another version of the interval timing task, a different patient population that from what I showed you previously, but they also have decreased 4-Hertz rhythms during the interval timing task. And here we picked a 12-second interval because we did this in mice. And what we found is that mice, if we deplete dopamine from the VTA, are missing this 4-Hertz rhythm. So why do I love interval timing? First of all, it's a great model of cognition, but it has translational validity in this context to understand the mechanisms of things that are malfunctioning in patients with Parkinson's disease who have dementia. Now, obviously, those patients have very complicated things going on, but this is a single thing that we can study. It's a thread to pull out. Some smaller end studies, but these are ongoing in my lab, and this is work again by Matt Weber, who's a postdoctoral fellow. Hopefully he'll be on the job market in 2023. He can do fiber photometry from the VTA dopamine neurons. Just to review, if you don't work at this level, you can express a sensor of calcium dynamics in the ventral tegmental area, and it gives you the relative signal of dopamine neuron activity, potentially relating to action potentials. In practice, it ends up being much slower, much noisier, but it does give you a pretty high fidelity readout of what the dopamine neurons are doing. And if you joined us just before the talk, what the dopamine cell bodies are doing might be very different from what the dopamine is doing at the presynaptic terminals, but that's a different story. But what we see is there's these 2 massive peaks in dopamine, 2 changes in the state. One is at trial start when that 4-Hertz rhythm would be triggered, and one is at reward, as you might expect. If you listen to NPR, dopamine equals reward. But this gives us some dynamics, because if we were going to manipulate the dopamine signal, and potentially 6-hydroxydopamine would manipulate all of this. It's like a sledgehammer or a nuclear weapon to the midbrain. So we wanted to get much more specific. In order to get more specific, we turned to optogenetics. Hold on. Oops. And what we did is we expressed halorhodopsin in the cell bodies in the ventral tegmental area, and we trained animals to do this interval timing task, and we could turn on the laser light, which should take the action potentials of VTA dopamine neurons to a very low level. And this gives us some temporal specificity because we are targeting this initial burst in dopamine that we see at the beginning of the trial. And so when Matt did this, and this is again n equals 3, because the data are rolling in, we see this increased variability in switch time. And so this is actually a much better control, because here we're turning the laser on randomly in the same trial, same within subjects design, but it's a very compelling demonstration that this seems to change the variability of the task. So in order to understand the next finding, I have to tell you a little bit about the neurons in frontal cortex. So this came through really blurry. So Eric Emmons, who's a grad student in the lab, now is an assistant professor at Wartburg, which is a teaching college here in Iowa, recorded from neurons in frontal cortex and striatum. And what he found was that single neurons tended to what we call ramp. So this means that over the interval, the neurons tended to increase their firing rate. And I could tell you all day about these kind of ramping patterns. If you have animals wait for shorter amounts of time, I'm sorry, this figure is so distracting. There's some issue with it. I can't figure out what's going on. I'm just going to delete all the white space and I'll make myself happier. Hold on. Okay, there we go. There's still some issue with the way I copied and pasted. That's all my own fault. So if we record for shorter intervals of time, the ramp ends up being steeper. So it seems to be that this is what we would argue is the key temporal signal in the frontal cortex. Now, you might say, oh, it's an interval timing test. The animals are moving. There are all kinds of problems in this test. True. In grad school, I recorded while the animal grabbed a lever and held it down for, seconds. And even when the animal wasn't moving, as far as we could tell, we saw these ramps. We really think they're the temporal code. We see ramps up, we see ramps down, and we see ramping in the striatum that depends on the frontal cortex, but that's a story for another time. And again, Eric showed this several times, but we didn't really believe this until Meredith Jazuri at MIT found this in the primate prefrontal cortex and striatum. So, this is kind of a highly vetted signal for time, although there are others who argue with me that there are other temporal codes, and we can talk about that if you'd like as well. So the reason I showed you those neurons is that, so Matt, in these optogenetic experiments, recorded from the frontal cortex. Here is a single prefrontal neuron, and it seems to have kind of this ramp down over the interval. Same neuron, different trials with the VTA inhibited optogenetically, the ramp seems to have changed. And so this is a really profound finding. It suggests that 2 seconds of VTA dopamine is sufficient to initiate ramping in the frontal cortex. So, it sort of localizes the dysfunction in a patient like Milton. Why does Milton have cognitive problems? Well, he can never initiate, never synchronize his frontal cortical neurons. So we know a lot about this ramping signal. So again, through behavioral pharmacology experiments, Crystal Parker, who's now an assistant professor at the University of Iowa, she put cannula in the frontal cortex and recorded, and if she blocks D1 dopamine receptors, or if she agonizes D1 dopamine receptors, the ramping seems to be exquisitely sensitive to prefrontal D1 dopaminergic tone. The first finding that basically kicked off my lab was we blocked D1 dopamine receptors and found that the animals could not time if they had no prefrontal D1 dopamine receptors. And I don't know if Roger knows this, but I came to Yale to work with Paco Marukish, who was very famous for her work of prefrontal D1 dopamine receptors in working memory, and really triggered me on this sort of D1 dopamine path. And I wonder what she would have thought if we can manipulate the D1 dopamine receptor system the way that, I'll show you subsequently the task. But partly because of Pat's work and work by Bita Moganam and Stan Floresco, it's well known that there's this U-shaped curve. So, Matt and I decided to put some data behind this. Paper in Behavioral Neuroscience that's coming out, we basically fit every study, and we were able to show that there is indeed a U-shaped curve of prefrontal D1 or dopamine, prefrontal dopamine or D1 dopamine receptor expressing tone by Cohen's D or working memory. And there's indeed a kind of, optimal level of dopamine at which this cortical system does things better. And this is very different than the striatum. In the striatum, if you increase, I'm sure there's a point where things break apart, but as you increase dopamine, you generally optimize striatal function up to a point. And then some stuff that we're following in the lab, again, this is new data and it's kind of n equals half a data set, but a new grad student in the lab, Hannah Stutt, is looking at the sex effects of it. So we, neuroscientists like me have been guilty of looking at male rats and mice for 100 years. So Hannah said, well, let's look at some female mice. Let's look at the estrus cycle. And so these are cumulative distribution plots of the time response functions. So previously I was showing you probability distribution functions. So the animals are supposed to switch around after 6 seconds. On the x-axis is the cumulative distribution of switch, and the y-axis is time. And we gave these animals sharing 23390, which is an old drug to block D1 dopamine receptors. So in males, it has one effect. And the point is, in females under diestrus, it seems to have the opposite effect, suggesting that there's this, really interesting intersection of sex effects, these estrogens, and interval time. So hopefully we will complete this data set, understand a little bit more, and have a more fleshed out story. And then Hannah's also studying amphetamine withdrawal. So if we, train animals up on amphetamine, give them amphetamine and withdraw them, males end up with a slower time response function, but females and estrus tend to be resistant to that. So these are, again, half data sets, maybe third data sets, but I just wanted to show you that, there's a lot of science that we can do with this setup. So I primarily am a single unit physiologist. I would rather sit at an oscilloscope and watch neurons fly by. I'd rather do that than just about everything else, and definitely more than most of my time, which is formatting grants. But we really try to record from these populations of neurons. And so in order to do that, we turn to single unit recording. And with optogenetics, we can use some of these techniques to actually tag the prefrontal D1 dopamine receptors. And this is where we think we would have gotten Pat's attention. So we use optogenetics to express channel rhodopsin in prefrontal D1 dopamine receptor expressing neurons. These are the postsynaptic neurons that are likely listening to the ventral tegmental area dopamine. We can pulse the laser, and the neurons expressing channel rhodopsin that have the D1 dopamine receptor will fire. And we can follow these spikes to the network, and we can find the neurons that are expressing D1 dopamine receptors, and we can also find neurons that are presumably firing after a synaptic delay of one synapse. And so we can use this type of technology to disambiguate the network. When we do this, the first thing is that, well, based on Crystal's data, which is here, we thought, oh, obviously the prefrontal D1 dopamine receptor neurons, obviously they're going to ramp. And we turned out to be, this is a case, previously I showed you a case where I was right, and here I'm, boy was I wrong. It turns out that if we look at the number of ramping neurons, the neurons expressing the prefrontal D1 dopamine receptor tended to be the least ramping population neuron. Whereas the neurons that were connected were intermediate, and the neurons that were not tagged, meaning that these neurons we think, although this is a viral strategy, so we can't rule it out, but they're not tagged by the laser, we think they're non-dopamine D1 dopamine receptor expressing neurons, tended to have the most ramping activity. So this is a really head-scratching result. And Young Cho Kim, who, I can't sing enough praises. Young Cho is a research assistant professor in my lab, did a postdoc in my lab, really developed a lot of the technical abilities of our group. He looked at the coherence of these neurons, bringing it back to that original finding that Rachel showed you. And what he looked at, so on the y-axis here is frequency, and the x-axis is time, and the color axis is spike field coherence. So if you see red, you see more spike field coherence, and you see blue, you see less spike field coherence. And he found that if you look at D1 neurons and their coherence with ramping neuron, he saw this massive 4 Hertz coherence phase locked to the Q. And this was not the case for D1 neurons with non-ramping neurons. So this finding, and again, it's one paper, one study, but it provides this kind of glimpse into how complicated this machinery is. And I'm going to give you a working model. And the only reason I give you a working model is that so you guys can shoot it down. There's Young Cho. All right, so here's our working. You have our prefrontal canonical network. And dopamine is released from mesocortical projections. These mesocortical projections trigger 4 Hertz rhythms. The D1 neurons are listening to this signal. They trigger these 4 Hertz rhythms, and they engage the neurons involved in cognitive control. So that's our working model. So just to review, dopamine is released, prefrontal D1 dopamine receptor neurons are listening to that signal, and they are going to get trained at 4 Hertz and gin up whatever cognitive machinery. Now, this is a general theory because it could work for working memory, it could work for oddball, Simon, timing, whatever cognitive flavor. And it explains why Milton is going into a nursing home. If he's missing, that mesocortical dopamine signal, he can never engage his cognitive control signals and never do the cognitive processes that's required for him not only to drive or work, but to interact with people, have conversations. But it also provides this enormous opportunity. And this opportunity is can 4 Hertz stimulation boost cognitive control? And so this is what we set up to test. Does 4 Hertz stimulation improve cognitive control in patients with Parkinson's disease? So the first thing we did, and this is Cho's work, is he took these here they are, the prefrontal D1 dopamine receptor expressing neurons expressing channel rhodopsin, and he stimulated them at 4 Hertz during an interval time test. He actually stimulated them at 2 Hertz because we didn't know better when we did this experiment in 2018. So here are animals doing this interval timing task, the BTA dopamine depleted in animals, and if he stimulates, if he stimulates at 2 Hertz at this delta frequency, he sees improved timing. And it isn't the case if he stimulates these neurons faster or harder at 20 Hertz. So potentially these neurons are firing more, there's more spikes, but it doesn't have this effect. So it seems to be a highly frequency specific effect. And then Matt turned to the ventral tegmental area. And again, what frequency is he going to stimulate at? He stimulates at 4 Hertz. And he's going to stimulate for that 0 to 2 seconds early in the trial. And again, the laser's only firing like 8 times, right? 4 Hertz for 2 seconds. So just to show you, the average switch time doesn't change if he turns the laser on. Again, these are individual animals, randomly chosen trials. He stimulates between 0 and 2 seconds at 4 Hertz. Doesn't change the average switch time. But if you look here, here's the cumulative time probability function. These animals the slope tends to steepen, meaning that the animal's variability tends to go down. 0 to 2 seconds at 4 Hertz, directly kind of what we hypothesized, and in line with other studies where you record from the VTA and the frontal cortex, there is coherence there between those two brain areas at 4 Hertz. So, a question we ask ourselves a lot is, does this matter? And so in order to ask that question, we turn to the operating room where we implant electrodes in the subthalamic nucleus. And when we implant these electrodes, we can fire them with whatever frequency we want. Of course, we had patients do the interval timing task. Here is 130 Hertz, our usual DBS settings. And if we fire the deep brain stimulator at 4 Hertz, we see this improvement in this highly restricted version of the interval time task. Again, this is work with Jeremy Greenlee. A lot of problems with this data. This is 7 patients, very small sample size. The task isn't exactly what we wanted. This is task we ended up to get to work. But it's compelling because it kind of points the way forward. And then let me also say that the STN is connected to the prefrontal cortex via the hyperdirect pathway. And if we record from the frontal cortex, we also see correlates of an increased 4 Hertz rhythm. So I didn't really, we published this result, but I don't know if I believed it until I saw this work from Catherine Scangos doing a Simon task. And I don't talk to this group because they stimulated at 5 Hertz. And if I did, I would have convinced them to stimulate at 4 Hertz. And what she found, or this group found, is that there was improved accuracy when they stimulated at 5 Hertz versus 0 or 130 Hertz. So providing an independent verification of this. Now, since then, I, of course, wanted to rigorously test this. So we came back to our Iowa. We did 18 patients on a multi-social interference task to try to see if we could get consistent effects. And I'm going to tell you straight up, these effects can be diverse. And maybe that's not surprising. The microanatomy of the STN really matters. There's a ventrolateral motor target versus a cognitive target. Maybe our 7 patients had more current in the cognitive domain. Maybe the task wasn't right. We really got to unpack this, but hats off to Rachel because, if we want to falsify it, we want to be the ones to falsify it. This 4 Hertz stimulation is not going to work out of the box. There's clearly a lot we're going to have to understand about it. I wouldn't even propose we do a clinical trial until we understand some of these key parameters. All right, so hopefully I've convinced some of you that dopamine is required for 4 Hertz cortical rhythms, controlling cognition. I've shown you that patients with Parkinson's disease have attenuated rhythms. Rodents allow us to explore the mechanisms, and that 4 Hertz stimulation has the potential to improve cognitive control in Parkinson's disease. 2 other things. So our NINDSP20 is essentially to do what I just suggested, that we need to understand sort of 4 Hertz rhythms in great detail. First project is an animal project using 2-photon and recording across layers in the frontal cortex. Project 2 is prefrontal interoperative neurophysiology, and project 3 is real-world EEG and MRI. These projects potentially will grow in scale, hopefully to an eventual P50 at Iowa, where we are working on it. This is my group. That's my Twitter handle, very important. And this is the book that I had the pleasure of editing with Roger. I'm really proud of it. It's really kind of the best, some of the best people in the world giving, the latest version of cognition and Parkinson's disease. I hope the book is all wrong in 100 years, but it'll be interesting to see what threads follow through. So anyway, thank you very much. Sorry I went too long. I think you wanted a 45-minute talk, but there you go.
Yeah, no, thanks. That was a lot of fun. So let me just start by asking a couple of questions. So first of all, what prefrontal region and what's the homologue in humans?
So this is a subject that I spent a lot of time hemming and hawing. So we are talking about medial frontal cortex, cingulate 2432. That is the node that people are going to sort of interrogate in default mode network. It's not dorsolateral prefrontal cortex. That is really hard to get at with scalp because it's kind of tucked, to the sides, and there's a lot of artifact there.
Right. Gotcha. Okay. Now, in your some of the human studies, I mean, did you interrogate drug on or drug off effects? I mean, is there any effect of replacement or?
You mean, so we have done, I'm going to go back here. So in the 4 Hertz, we looked on versus off, and we did not see any effect of dopamine on this 4 Hertz rhythm because of the reasons that I showed you. I think it's because of this. I think it's because of this U-shaped curve. You got to hit the right level of dopamine. And so, more dopamine doesn't always help you. And actually in Arun's papers, he showed that, I think it was only 9 patients, but other people have done this since then. These 4 Hertz rhythms don't really improve with levodopa. Yeah.
Well, I think you're right. I mean, the dome-shaped response curve would be the explanation, but in part, in addition, the other factor would be that it's been suggested that dopamine clearance is different in cortex than the striatum. It's more dominated by COMT. So which means there could be an effect of the COMT polymorphism that affects COMT activity. I suspect you need a large group of subjects to disentangle that.
But Fultini, the guy in Britain, there's a group in Britain that has this cohort. Trevor Robbins' group, right? I think they have.
It's a fairly common polymorphism.
We should think about, I'm trying to think about if we could sequence our patients,
you could probably, I'm sure there's an easy PCR assay for this. That would be my guess.
That's a really interesting concept.
And just before we get other people ask questions, the other thing is, I think, with the estrous cycle stuff, you probably already know this, but Jill Becker here in our psychology department did a ton of studies on estrus and various aspects of striatal function.
Actually, Hannah is our grad student who's really pioneering the way on this. She's turned me into a Jill Becker fan. So I'm I need to talk to her at some point. She's done just amazing work. I've only read a fraction of her papers, but it's just amazing the stuff that she's done.
A fraction of her many papers, right? Right. So okay, I'll stop hogging the microphone here. So, any other questions? So, okay.
I'll throw one out there. This is really interesting. We looked at the correlation of kind of like delta rhythms and reaction time and find out a good correlation in thalamic neurons. I'm wondering if since there is a phasic relationship, you could think about turning around these tasks. Let's just take reaction time and do like a closed loop control, and instead of asking the rat to react with the light or something, kind of take control of the stimulus or the sound, whatever that is, and have that closed loop with the ongoing 4 or 5 Hertz rhythm, and see if like that relationship is really true. I don't know what your thoughts are, like where you're going with any closed loop controls.
My thoughts are, those are like our next several grants. But so Young Cho Kim, who's on this call, is a technical, unbelievable. He's gotten down to 13 millisecond latencies with our closed loop control systems. So we can actually sample and stimulate in and out of phase with these rhythms to do exactly that. I think that you might be able to use closed loop technology to boost the rhythms, and you don't need to boost them all the time. If you boost them at key moments, that might be enough to engage these neurons that are not part of the picture. But that principle of using behavior or neural activity, maybe I'm completely wrong. I'll give you an example in this talk of where, I had some evidence behind me and where it's completely wrong, right? So closed loop will enable you to test those hypotheses in a much quicker way. And so we're trying to double down on that. Getting it to work in humans is a little bit more of a trick because there are all kinds of appropriately lockouts. Brooke Yeager in my lab is doing transcranial alternating current stimulation, and she's trying to individualize the stimulation to the frequency, native frequency in patients. That's had some mixed results, but potentially these themes are what we need to do if we're going to personalize stimulation. Our brains are different. There's no reason why our rhythms are different in it, and it's a hypothesis that if we come up with a principled approach to thinking about that problem, we might be able to optimize some of these neuromodulation interventions, and we might also be able to use feedback. I referred to like continuous measurements. It might be we're able to come up with sort of kind of a stimulation paradigm that improves some of these biomarkers and then improves function. But those are now I'm like grants that I haven't even dreamed of yet. So great question.
Can I ask you about your focus on the 4 Hertz rhythm? Because number one, when I looked at your spectrograms, I thought there was a washout pretty much across all frequencies. And of course, you're driving with those low-level non-cognitive tasks, 4 Hertz rhythm. So those are really popping up. But for me, in my world, the 4 Hertz rhythm in a subject that is at least not in slow wave sleep really exists largely to support a cross frequency coupling, usually to higher frequencies and usually to gamma. So I'm just wondering whether that sole focus on 4 Hertz really is justified, and if you were really tapping into something more cognitive, whether you would see that actually the entire spectrum is disrupted and phase amplitude couplings to higher frequencies are gone. And that's really the issue at the end of the day that I would think is going to correlate precisely with the cognitive decline in these patients.
So I love your question, Martin. It's very insightful. I have a long answer, but here we go. So first of all, we do see in our spectrograms a couple decibel enhancement of 4 Hertz activity. I don't see that washout that you referred to here, but it's kind of a consistent finding. It's short, it's Q triggered, but it is largely consistent and it's baseline normalized. But we can go to the bank on this kind of rhythm, and it predicts future behavior. And it actually in all of our papers, we compare it versus the beta band, and in some cases versus the gamma band. Among the bands that we analyze, this is the most predictive. So the R² values are much higher than those ones. But your point about phase amplitude coupling is very insightful, because it is possible that there's one rhythm and there's another rhythm that nests in that rhythm. And potentially the interneurons actually play a huge role in that process. And I think Young Cho and I have disagreements about that, but Young Cho is actually setting up to record from these interneurons. I would bet that the gamma rhythms are multiplexed from these interneurons on these 4 Hertz rhythms that are, I think, pyramidal cell. But that's a hypothesis. We want to test that. But your point, though, at the end of the day, we want to modulate the network, and we have stimulated at higher frequencies. We've stimulated at 20 Hertz. We haven't used sort of better options to go to the gamma Hertz, because I'm a little skeptical of those options, but maybe I don't need to be. But if we use Cheetah or something, we might be able to test this. If we go higher in our frequency bands, we don't see improvements in our behavioral tasks. And then the last thing I'd say is, when you say non-cognitive, I would quibble with that. We have data to actually show that our task performance, and I'll show you this data, is cognitive. If you, and not just our data, there's a guy named Brown who basically had patients do timing tasks and work memory tasks, and there's absolutely interference with these tasks. I don't know what a non-cognitive task is, but at least in these low-level timing tasks, we think that we are engaging some of the same circuits that are involved in these further, and we have some data to speak to that. But I think your concept of phase amplitude coupling is awesome. We haven't thought about it as carefully as we need to, and we certainly need to go after that. So you're right about that point.
Okay. I think we're at the end of the hour. So, so Kumar, thank you very much for a really fun talk. And we'll see you and your group in a couple of months, I think, actually.
Yeah, right. I think Rachel and Matt are going to be speaking of that thing. So
yeah, that'll be a lot of fun, actually. So, and thanks everyone for attending and for Martin and Matt for very interesting questions as well. So thanks very much. Okay.
Thank you. Last thing, if you have other questions, feel free to reach out to
Yeah.
me. If I can help in any way, in anything you're doing, please let me know.
Okay. Thanks very much, Kumar. Great to
Thank
see
you.
you.